Okay, as a summary to the previous material about chemical neurotransmission, you should now appreciate that chemical neurotransmission is the foundation of psychopharmacology, and that does not necessarily mean that we support the view that everything psychological is biological in origin. We are talking about the relationship between psychology and biology. And we have biological, I would say, reflection of psychological events. We are not talking in that way of making the biological aspects as the origin of psychological problems. No, that does not. Our understanding of these subjects does not necessarily mean that. So, there are many neurotransmitters, and all neurons receive input from a multitude of neurotransmitters in classic presynaptic to postsynaptic asymmetrical neurotransmission. What also, what we've talked about also in this chapter, that presynaptic to postsynaptic neurotransmission at the brain's trillion synapses are key to chemical neurotransmission. But some neurotransmission is something else other than classical neurotransmission. So we have this retrograde form of neurotransmission from postsynaptic neuron to presynaptic neuron. And other types of neurotransmission, such as a volume neurotransmission that actually does not need or require any synapse at all. You also should have an appreciation for elegant, if complex, molecular cascades precipitated by a neurotransmitter, with molecule by molecule transfer of the transmitted message inside the neuron receiving that message, that eventually altering the biochemical machinery of that cell in order to act upon the message that was sent to it. Thus, and this is important, just note that, concentrate with me, the function of chemical neurotransmission is not so much to have a presynaptic neurotransmitter communicate with its postsynaptic receptors. No, it's not as simple as that. It is actually to have a presynaptic genome converse with a postsynaptic genome, DNA to DNA, presynaptic command center to postsynaptic command center and back. So it's about the genome. The message of chemical neurotransmission is transferred via three sequential molecular pony express routes. First, we have a presynaptic neurotransmitter synthesis route from presynaptic genome. Let's just understand it like this. We have presynaptic genome that produce the presynaptic neurotransmitter. Okay, so the synthesis of that neurotransmitter and the packaging of that neurotransmitter and supporting enzymes and receptors. Okay. Then, second, a postsynaptic route from receptor occupancy through second messengers all the way to the genome, which turns on postsynaptic genes. And third, we have another postsynaptic route starting from the newly expressed postsynaptic genes, transferring information as a molecular cascade of biochemical consequences throughout the postsynaptic neuron. It should now be clear that neurotransmission does not end when a neurotransmitter binds to a receptor, or even when ion flows have been altered, or second messengers have been created. Events such as these all start and end within milliseconds to seconds following the release of presynaptic neurotransmitters. So that's not the ultimate thing of the ultimate goal of neurotransmission. The ultimate goal of neurotransmission is to alter the biochemical activities of the postsynaptic target neuron in a profound and enduring manner. Since the postsynaptic DNA has to wait until molecular pony express messengers make their way from the presynaptic receptors, often located on dendrites, to phosphoproteins within the neuron, or to transcription factors and genes in the postsynaptic neuron cell nucleus. It can take a while for neurotransmission to begin influencing the postsynaptic target neurons' biochemical processes. The time it takes from receptor occupancy by neurotransmitter to gene expression is usually hours. Furthermore, since the last messenger triggered by neurotransmission, called a transcription factor, as we said, that only initiate the very beginning of gene action, we call it early genes, it takes even longer for the gene activation to be fully implemented via the series of biochemical events it triggers. These biochemical events can begin many hours to days after the neurotransmission occurred and can last days or weeks once they are put in motion. Thus, a brief puff of chemical neurotransmission from a presynaptic neuron can trigger a profound postsynaptic reaction that takes hours to days to develop, and that can last days to weeks or even a lifetime. Every conceivable component of this entire process of chemical neurotransmission is a candidate of modification by drugs. That's why we talk about them and about these processes in this material. Because most psychotropic drugs act upon the process that control chemical neurotransmission at the level of neurotransmitters themselves, their enzymes, and especially their receptors. Future psychotropic medications will undoubtedly act directly upon the biochemical cascades, particularly upon those elements that control the expression of pre- and postsynaptic genes. Also, mental and neurological illnesses are known or suspected to affect these same aspects of chemical neurotransmission. The neuron is dynamically modifying its synaptic connections throughout its life in response to learning, life experiences, genetic programming, epigenetic changes, drugs, and diseases, with chemical neurotransmission being the key aspect underlying the regulation of all these important processes. That's why we need to understand them in details. So that's it as a summary.